Okay, so we're going to look at aerobic respiration first, and this section is going to be glycolysis. So I'm going to look at each of the stages independently. So first of all, aerobic respiration, glycolysis. So I'm going to do my diagram here on the right, and then the key facts that we're going to need to know about it on the left. Okay, so I'm going to keep my notes and my colors very consistent whilst we're doing all these respiration and including photosynthesis as well. So this is well worth copying out a few times. I'm going to suggest to you at the end you're going to make a skeleton diagram of the empty diagram, and then you can use that as some revision purposes. First up, we start with aerobic respiration, glucose. I'm going to put these in boxes. Now, glucose is going to break down into something called glucose phosphate. And to do that, we need to put in some ATP, and this is putting in ATP. So ATP goes in and ADP comes out. We don't need the inorganic phosphate because we're adding the phosphate to this molecule, so it's become glucose phosphate. So far this reaction is helping you out because the names all coincide. Next thing is that we're going to add some more glucose, and we're going to make hexose bisphosphate. How do we do this? Well, the bi that we've got here, almost like biphosphate, two phosphates. Well, we do it again. We add some more ATP. So this is ATP going in, ADP coming out. And this first half of glycolysis is known as phosphorylation. We're adding phosphate. Okay, so far we've used 2 molecules of ATP, which is exactly not what we started out to try and do. We want to try and generate some ATP. This is respiration. But what happens next is going to yield us some ATP. So I'm going to draw this in parallel, two of exactly the same things happening, because this is going to split into 2 molecules. So we can come out here, and equally we can come out here. And this is a molecule you've come across in the Calvin cycle as well. This molecule here is triose phosphate. These are exactly the same. You can write 2 times, but I find it much easier to visualize in terms of remembering all the other reactions going on if they're drawn out side by side. Okay, so how do we get some ATP out of this? Well, finally we convert triose phosphate and we get pyruvate or pyruvic acid. And this is the reaction that yields us some ATP. So between here and here, same on both sides, we have 2 ADP and PI going in, and we have 2 ATP coming out. And that's obviously going to be the same on each side. So here we've got 2, and we've got 2, that's 4, and we've only put in 2, so we've got a net yield of 2 ATP. Now, at this point, we start doing some other things as well. So here, we also take, this is a coenzyme called NAD. You've met NADP in plants. This doesn't have a P, it's not a plant. It's going on, well, this is where the analogy breaks down slightly, but let's say this is respiration. Plants obviously respire too. NADH, or reduced NAD. And exactly the same. And there's one of those on each side. Okay, so we're going to also look at the carbon atoms because it's going to make this much easier to understand. We should know by now glucose has got 6 carbon atoms. Well, we've not added it or taken it away in any carbon atoms here. So also 6 carbon atoms. Hexose tells us it's got 6 carbon atoms. Triose, well, triose should be pretty obvious to you these days as the amount of science and biology you've done. We've got 3 carbons, and we've got 3 carbons. 2 times 3 is 6. That's perfectly, it all adds up, and we've not lost anything. 3 carbon atoms. 3 carbon atoms. Now, what is going to happen to these products is that eventually, oh, not very nice, these are going to go into a mitochondrion. And this is what happens to pyruvate. They actually go into the matrix, which is where link reaction and Krebs cycle take place. people frequently forget that this happens in the cytoplasm. And this part of the reaction here, this is oxidation. We're going to look at what oxidation and reduction are in some key terms, but basically it's lost hydrogen and electrons. So the trisphosphate has been oxidized to form pyruvate. The NAD has been reduced to form NADH, or reduced NAD. So you can also refer to these as redox reactions. When something is oxidized, the other thing is reduced. So what are the key facts that we need to know? Glycolysis takes place in the cytoplasm. It does not take place in the mitochondria. I'm even going to put this in red. You could say that glucose is too big to cross the mitochondrial membrane. You could say that there are no transport proteins to move transport across the membrane. Both of those things would be fair to say in an exam, which has come up on the exams in the past. It's anaerobic. There's no oxygen involved in any of these. I'm going to just write here that the net yield is 2 ATP and 2 NADH, or reduced NAD. Okay, so glucose is phosphorylated in the beginning by adding ATP. And then it is oxidized. So the chain of reactions here is glucose, glucose phosphate with one addition of ATP, glucose phosphate, hexabisphosphate with the addition of one more ATP. We can then say that triose phosphate is oxidized to form pyruvate. And pyruvate is then actively transported into the mitochondria for the link reaction. Okay, so to revise this is complicated, detail rich. There's not that much information that you need to know, but you basically need to know all the little numbers and figures, because knowing these carbons are going to really help you out later on. So the skeleton diagram I'm talking about is basically this exact diagram. You can put the curly arrows in, the straight arrows in. You can give yourself as many clues as you want to begin with. I normally draw it out. I'll then photocopy it or scan it and print it, however you want to do it. And then you can fill in these bits of information. And the better you get at it, the less information you need to put on it, until eventually you can just sketch this out. Glucose, arrow, glucose phosphate, boom. You can stick all that information on there. I could probably do that roughly, that I didn't want it to be replicated, in 2 minutes easily, probably less than that. So it will take a little bit of learning, but go through it. Basically, you're covering it up, what section's which, and then you're going to learn it much faster from there.